In this lecture, we will zoom in and look at some specific mechanisms of gene regulation by epigenetics. But before we do that, let's have a quick overview of what epigenetics mean. Epigenetics is the study of mechanisms that lead to changes in gene expression that meet three criteria. One, they can be inherited. Two, they are reversible. And three, they do not involve changes in the DNA sequence. So when we talk about epigenetic inheritance, we are talking about changes to DNA structure, but not sequence, that can be passed from parent to offspring. There are several types of molecular mechanisms that fall under the umbrella of epigenetics. These fall into five categories as shown here. We will explore a few of these in a bit of depth, but know that from the outset that we could spend a ton of time on these mechanisms if we chose to. Let's begin here by talking about chromatin remodeling. So chromatin remodeling, which we have previously introduced, involves changes to the structure of chromatin in our nucleus. Recall that our basic chromatin structure involves our DNA wrapped around histone octamers. In addition, this structure also includes non-coding RNAs, which can be important for targeting which genes are selected for remodeling. Chromatin comes in two varieties in our cells: euchromatin, which is transcriptionally active and less compact, and heterochromatin, which is highly compacted and generally not transcriptionally active. Additionally, these occupy different regions of our nucleus, with euchromatin generally found in the central portion of the nucleus and heterochromatin generally found along the periphery. Existence of our DNA as heterochromatin has several roles, including gene silencing, prevention of transposable element movement, and prevention of viral proliferation. These are all important mechanisms for controlling which genes get expressed at a particular time or in a particular cell type. Our heterochromatin falls into two types. Constitutive heterochromatin is always heterochromatin in every cell type. Facultative heterochromatin can actually be less compacted euchromatin in some cell types. So let's start with constitutive heterochromatin's characteristics. These are found at the same place in every cell, generally in highly repetitive sequences close to the centromere and telomeres. This chromatin is highly methylated and often has specific histone modifications that help it maintain its high compaction. Facultative heterochromatin is more flexible, and in some cell types or stages of development is found as heterochromatin, and in others, euchromatin. This is generally found between the centromere and telomeres. When these genes are found as heterochromatin, they tend to be methylated at the CPG islands in the gene regulatory regions. What's cool here is that chromosomes will generally follow the same pattern of constitutive and facultative heterochromatin from mother to daughter cells. The formation of heterochromatin in our cells follows three basic phases. In nucleation, a short chromosomal region is bound by modifying enzymes and remodeling complexes. In the spreading phase, the euchromatin is compacted into heterochromatin in this short chromosomal region. Lastly, in the barrier phase, the spreading stops once it reaches barriers between heterochromatin and euchromatin complexes. As mentioned previously, this pattern of heterochromatin is maintained across cell divisions. What this means is that when our cells divide in our bodies, the gene silencing patterns stay the same, and our two types of chromatin are maintained. This is great, as we want to keep our cell types the same pre and post cell division. The mechanisms that maintain these epigenetic marks are shown here and include DNA methylation, maintained through maintenance methylation, histone modifications that recruit chromatin remodeling complexes in daughter cells, specific DNA polymerases that can recruit chromatin modifying in complexes, and some aspects of higher order structure, which we won't dig into here. The last topic here is the role of heterochromatin in human disease. Maintaining proper gene expression patterns in multicellular organisms is important, and when these mechanisms fail, they can lead to phenotypic consequences. ICF syndrome is due to a mutation in DNA methyltransferase, which is responsible for maintaining the methylation patterns in a cell. This can lead to a host of phenotypic consequences, including immunodeficiency, facial anomalies, and centromere instability. Roberts syndrome is due to mutations in the enzyme acetyltransferase. This leads to growth defects, craniofacial abnormalities, and limb malformations. Recall that acetyltransferase is responsible for acetylation of chromatin, which loosens the histone DNA binding complex.